personally introduce today's moderator, Dr. Mark Baker. Dr. Mark is a current professor in Macquarie University in Sydney, Australia. Mark is a pioneer in proteomics research, was involved in the organization of International HUPO very early on. He was president for the US International HUPO from 2015 to 2017, and currently is the chair for Human Proteome Project. And he held several faculty positions in Australia and certainly on editorial board for multiple scientific journals. And he's currently senior editor for the journal Clinical Proteomics. And besides his academic achievement, Mark was also the director and founder for several biomedical centers and biotech startups. And so I think it's quite appropriate today for him to moderate and interview Parag. Without any further ado, let me welcome Professor Mark Baker.
Thank you, Andy. It's a real privilege to be involved in these sessions, in particular, given that it's before 6 o'clock in the morning here in Sydney, Australia. It's really a wonderful opportunity for us to get in at the ground level now and hear from a colleague that is developing new technologies with regards to the discovery of the proteome. So it's I'm having a little bit of trouble with the screen, Andy, so I'll try and get up my notes. I'm having a little bit of trouble getting them up. So let me just close the chat a bit and see if that helps. Okay, that seems to help a lot. So the session today has actually got two main parts to it. The first is for us to discuss the future of next-gen proteomics. And the second is to explore the opportunities and challenges that are associated with commercializing emerging technologies. And it's really quite a great opportunity for us to do that, given that Parag and his colleagues have only just recently launched their company Nautilus Biotech on the NASDAQ, with a number of very prestigious venture capitalists getting behind it, including some of the venture capital companies that are familiar to many of you, and we might explore them later on. We do have opportunity for questions, so please use the software. If you wanted to ask questions directly of Parag and myself, please email us later, and we'd be more than happy to interact with you and to add materials. So Dr. Parag Mallick is a lifelong member of our proteomics community. His original training was in both computer science and biochemistry, quite an interesting mix while he was at Washington University. He obtained his PhD at UCLA in chemistry and biochemistry, where a lot of us have obtained our PhDs, and he worked there with Dave Eisenberg. And after that, he completed his postdoctoral training with Pioneer and the current Human Proteome Project Scientific Advisory Board Chair, Rudy Aebersold, at the Institute of Systems Biology in Seattle. And there he worked on the basic science side of defining the biophysical origins of proteotypic peptides. Parag also did some early work in proteogenomics and proteomics-based biomarker identification. So his skill set covers quite a broad range of those technologies that are currently being incorporated into the mixture of things that are needed to commercialize companies. He's now an associate professor at Stanford University, and he uses there a mix of quantitative proteomics, machine learning, AI, nanotechnology to perform systems biology studies, particularly on cancer progression and aspects of cancer in the Canary Center there at Stanford. Additionally, he was the initiator and PI of the Proteome Wizard project, and his group has been at the forefront of open source multi-omics data analysis methods for some time. Beyond academic pursuits, Parag has recently co-founded and is the chief scientist of Nautilus Biotechnology. It's a proteomics platform technology development company that he founded 5 years ago. And I think the story of the growth of this company is going to be one of the interesting things we hear about today. They've been working to create a large-scale single molecule platform for the quantifying of the human proteome. This is an extremely ambitious task that they've taken on. And their mission is to develop that novel platform for proteomic analysis that will help democratize access to the proteome, thereby enabling fundamental advancements across human health and medicine. So it's with great pleasure that I get to welcome you, Parag, and I look forward to our discussions today.
Thanks so much, Mark. It's really great to be here, and it's great to see so many friends amongst the attendees. So I'm really looking forward to our chat today.
Fantastic. Congratulations to you, Sujal, and all the team on the launch of Nautilus on the NASDAQ under the ticker NAUT, N-A-U-T, and also for attracting a really impressive list of investors like Jeff Bezos' Expeditions and Paul Allen's Vulcan Capital. You know, we have a warm friendship that's gone back many years, and we both watched proteomics develop since its inception. So I wonder if the start, if we can go back a little bit in time historically, and could I ask you just to explore what you think we've learned from the experiences of the past of trying to take proteomics into a commercialized setting?
Yeah, that's an interesting question. I think we've seen a couple of different classes of companies addressing different pieces of the ecosystem. A whole set of companies developing novel core instrumentation that are, of course, essential to everything we do. A set of companies that are really building on top of those core platforms with different additions and modifications, different chromatography, ionization sources, those kinds of things, as then a lot of effort in different sample preparation workflows and technologies to help along the way with better quantitation or more reproducibility, different fractionation techniques, and software then for helping interpret all of this data that is just complicated, and then on the sort of the back end, really a set of companies that are using proteomics to execute workflows for either therapeutic discovery or as part of contract services. And I think what we've seen is that those companies that have really just succeeded tremendously had a really close match between solving an important problem in an effective manner and at a time when that solution was critical. And so I think those two aspects. We've also seen that we've had some failures where the markets evolved. That a disruptive technology came around and obviated the need for something, and there also have been other cases where we've had some really just cool science, but that didn't have a huge market need, or the market need was so narrow. And so that match between what is needed and what we can offer, I think has really been a huge important point of focus. And it's also been a huge focus for the funders, that we've seen ebbs and flows in the excitement from the funding community with regards to proteomics. And a lot of the questions have been, okay, well, is this important? Who wants to do this? Why do they want to do it? And us as a community doing an improving, better job of elaborating the need throughout the biological ecosystem.
Yes, great point. Failures and setbacks give us a great opportunity to move things forward. Can you give us some examples of where those you know, just in terms of the types of experimental approaches that have led to us moving forward because of those setbacks or failures.
Yeah, I mean, I think just even within the mass spec ecosystem, we've seen just tremendous evolution. I remember, you know, from our early days in Rudy's lab, we were so thrilled with what we had, and now, if you put us in front of today's instrumentation, our minds would have been blown by what was available. And so I think we've seen those successes of just iterative advances over time. Yeah. But we've also, I think, there have been some technologies that we've really struggled to get past, and in particular, some of the hype in the very early days of proteomics that I think we're still struggling as a community to get past, and is a really important lesson for us to be aware of in this renaissance that we're in right now. Of making sure that we accurately describe what we're doing and we don't overhype things too much.
Yes, you mentioned a renaissance, and the fact that overhyping and under-delivering is a potential risk that we might fall into again. I wonder whether you might be able to give us sort of some reflections that you've had on that renaissance, on that inflection point. I see it as an inflection point where proteomics has resulted from widespread analytical and computational tech advances, standardized workflows, better data rigor and reproducibility, and public sharing of data. And then you combine that with what I think is a pretty strong movement that's acknowledging genomics alone doesn't provide sufficient depth or sufficient insight in biological experiments to answer everything. So can you provide an overview maybe of the major solution approaches that are being used to tackle proteomics experiments at this point in time?
Yeah, absolutely. I mean, I think there are three really big classes that we see, and of course, the folks here are likely familiar with many of them. You know, I think on one hand, we have the mass spectrometry-based approaches, whether targeted or broad scale, DIA, others that are a whole canon of techniques. Then we have the affinity-based approaches, so the arrays, the aptamers, the Olinks of the world that are really targeted approaches for looking at an increasingly large number of analytes. And then I think we have a third class, which is just emerging today, which are companies like Nautilus, which are alternative platforms, single molecule platforms. And I think we're just at the start of that new wave of emerging platforms.
Yeah.
And I'm curious, you know, if you'd asked that question, if I'd asked that question to you about 15 years ago, you probably would have had a quite different answer. You might have said, something about DIGE, and but I'm curious, what have you seen as the evolution of the core technologies in the field for over the course of the last 15, 20 years?
Very interesting point. I remember when a very early Australian company was using Kamasi Brilliant Blue Stain 2D gels, and at that stage fluorescence was all the rage worldwide. And I remember talking to the CEO of the company suggesting that a movement towards using fluorescence would be a great idea, but to tie that with peptide mass fingerprinting and to tie up a mass spec end to them. And that was, believe it or not, only 2003. So we've, as you've said, we've come an enormous way in quite a short period of time, just over a decade, or a decade and a half. The other things that I think have happened through things like the Human Proteome Project have been this reliance on high stringency data, on making sure that mass spectrometry and any orthogonal technology that we use to back up our initial results are used with very high stringency, that we, again, don't try and overclaim, but over-deliver on projects. So I see those two things, and certainly the sharing of data has been fantastic, and the US has led the way. US and Europe have led the way there with both, say, the CP tax sharing of data, and also the FAIR principles that have emanated originally out of Europe, but are now adopted by most people. And the major changes, I think.
Yeah, you know, I certainly, when we talk about data sharing as a catalyst, that is something that 20 years ago is something that we were actively thinking about in creating the MZXML standards and ultimately MZML and some of these other things that, of course, ProteoWizard has been part of. But it really led to a transformation in both the tools that were accessible for interpreting the data, the community of folks developing tools to analyze and interpret data, as well as in just this point about proteomics not being limited, and interest in proteomics not being limited to proteomics practitioners, to folks who have a mass spectrometer. I think that has been a very important change that we've seen, and through things like CPTAC and just the number of people who are downloading those data sets. And that's really led to a renewed interest and excitement alongside these seminal papers, things like the Human Proteome draft from a few years ago, and then the highly validated draft from this year that you, of course, were senior author on. And so I do think that it's been a convergence of a couple things. One, like you said, the recognition that there's a lot of value and importance in the proteome, that the genome's not going to get you everywhere. Which, of course, to folks in the proteomics field, they've been saying that for a while. But to the broader community, that broad recognition, you know, the technologies themselves have matured a lot. Our understanding of how to use the technologies, and then just the analysis and ecosystem. I think those have all converged at a really unique moment today that is very special.
Well, the proteomics renaissance, I'm looking forward to seeing where you take it. Parag, a recent GeekWire article, or a journal that I read quite a bit, mentioned that Nautilus was developing a brand new way to analyse the proteome. I wonder if you could give us some more detail on how you see Nautilus's novel platform working, and because many of the proteomics practitioners here are still using MS and are not all that familiar with the strategies that you've taken. So could you provide us an introduction to the aims, the technology, and the directions that are specific to Nautilus's technology?
Yeah, for sure. I'm not going to take too much time to go through it, but just to highlight the core principles of the approach.
It's 3 to 5 minutes, so yeah. Yeah,
so I think there are really 3 core pieces to Nautilus's platform that work closely together to enable our larger goal of measuring the whole proteome in a way that is relatively straightforward for the user, so that we can democratize access to the proteome. That is, we have an internal saying, sort of a slogan at Nautilus, which is anyone who wants a proteome gets a proteome. And so the first part of the platform is really thinking about the problem of how do we detect proteins a little bit upside down. When you think about your standard protein assay, like an ELISA, you immobilize a lot of antibody on a surface and then try and grab things out of solution, and you do that in bulk, and then you take your plate and you throw it away. On Nautilus's platform, we quite literally take that and flip it upside down. And our early goal was to say, well, we know we need something that needs to be extremely sensitive. We don't have PCR, so we can't copy or amplify things. So if we want to get to those rarest proteins, we really need a platform that can actually measure down to a single molecule. And so the way that we address that is by taking our sample and immobilizing it on a giant surface, a massive array. You just picture in your head a big chessboard, where we're going to take every single molecule from that sample and drop it on its own individual cell of that giant chessboard. And so, which is non-trivial to do, but that's the first step in our sample prep process. It is an intact process. We don't digest the proteins, which is important for both looking at proteoforms as well as for dynamic range concerns. So once you have those proteins immobilized on the surface, the problem of proteomics, of figuring out what's there and how much, fundamentally changes. Because at the single molecule level, identification of proteins and quantification of proteins become the same thing. If I can figure out what each of the molecules are, even though they're distributed in a variety of different places and disorganized, I get my quantification just by counting. So the second question is, all right, how do you actually identify those proteins? It's important to note that those proteins are glued down to the surface. They're not just adsorbed, they can't go anywhere. And so that allows you to interrogate them over and over and over and over and over again. So now to ask the question of, okay, I'm looking at coordinate 2,1, what protein is this? One approach that you might figure out what that is by flowing in a targeted antibody, something you could buy from a catalog today. You fluorescently label it. We have a special label that we use. Flow it in, it'll bind, and you'll say, oh, look, there's an EGFR at coordinate 2,1. So that's one approach. But and it works great, and it's a really phenomenal way to start digging into proteoforms, because you can sit there and plunk away at a given molecule over and over and over again and look at different modifications. But for scaling to measuring the whole proteome, it would take tens of thousands of cycles, and you would need great, perfectly specific affinity reagents for every protein in the proteome, and that seemed a challenging problem. So instead, what we do is something a little bit different. We're always complaining about affinity reagents cross-reacting, and so we recognize that the instrument maybe could work a little bit differently. So what it allows you to do is it allows you to flow affinity reagents, do a detection, wipe it off, flow an affinity reagent, do a detection, wipe it off, and do this iteratively over and over and over again. So the recognition was, well, what if instead of trying to build affinity reagents that were perfectly specific to single proteins, we instead tried to build a new class of affinity reagents that were intentionally highly cross-reactive? So things that target short epitopes, for instance, 3-4 amino acids long, those are going to show up in many, many, many proteins. But what you can do is label them, flow them into the flow cell, they'll bind to some set of proteins, wipe them off, come in with another one that again binds to a multiplicity of proteins, and build up a set of information for each individual molecule. A little nugget of information that you get from each probing. So while each probe itself isn't specific, it gives you a little nugget.
So each probe would give you some sort of either family typic or epitope specific data for each point that is on the star map that you're creating. And when you create the star map, say the first run through, you will end up with the Northern Hemisphere star map. And then with the next probe, you'll see a different star map, the Southern Hemisphere star map. And that's the nth degree, depending upon how many reagents you use.
That's right. And so that over time, you might look at that coordinate 2,1 and you'll say, okay, well, it bound to probes 2 and 3 and 85 and 92. And then you take that digital information. And this was really the third key insight to the platform, was saying, well, we typically use machine learning in biosciences post facto. We use it after we've collected our big data sets and we're doing differences between cases and controls. But if instead we brought that into the measurement itself, so instead of limiting ourselves to signal processing, image processing, if we actually use that machine learning step to take those patterns of binding events and ask the question, what protein is compatible with that pattern that we've observed? A very surprising and counterintuitive result was that you didn't need terribly many touches with these special probes in order to uniquely identify each protein. And that's really the heart of the platform, is recognizing that with this more fuzzy sensing approach, that you could potentially uniquely identify, 98% of the proteins in the proteome.
So you're doing fuzzy logic proteomics in a way.
Yes, that's right. That's right. And so, but it relies on a very special combination of hardware in terms of this nanofabricated chip, in terms of the imaging instrumentation, which is very specialized. It also relies on this very special biochemistry wetware in terms of these probes and the ways that we immobilize the proteins on the surface. And then it requires pretty sophisticated data science and software. All of those three working very closely together, ultimately to provide a system that looks a lot more like a DNA sequencer than it does look like a mass spectrometer.
Is that what it looks like on the bench, for example? Is that how you envisage it appearing to the researcher?
Yeah, absolutely. So that's definitely the goal, is that it is something that, just like a DNA sequencer, people walk up to, they drop a flow cell on, they press a button and hit go, and then out comes a proteome.
Okay. So one of the really interesting things when you take something that's so disruptive as this, and have the idea, I gather you had the idea originally in an academic environment, and then take it to a commercial environment, is the commercialization steps, all those series of things that have got to be done that involve protection, that involve making sure that the value proposition is secure, and then disclosing it to the public effectively by saying, okay, we've got a protected invention, and then we're going to commercialize it. So you must have learned a hell of a lot of new things taking this from the academic lab to a commercialized product and launching it on NASDAQ. Would you like to, I know there's probably a lot of things you've learned there, but are there any keys that you think that this sort of audience would really take home from that experience that you've had?
Yeah, well, so I think certainly the first question that I would, where this all came from a crazy brainstorm on a weekend, that literally I woke up one day and had this crazy thought, and so then the question was, what is the right vehicle for getting this out in the world? Is it through my academic lab? Is it through licensing it to a larger company, like one of the big life science vendors?
Yeah.
Or was it through a startup company? And so as I went through the process, it became clear that each of these vehicles have different strengths and different challenges as well, of course. So, and I see there are several questions here about funding, and when I started off, it was clear that protection was very important. So very early on in the process, I did reach out to our IP counsel, Wilson Sonsini, and asked them for described this idea, and they said, "Wow, that's a really interesting idea. Let's get that protected." I was very transparent with Stanford's technology licensing group the whole way, describing this, sharing with them everything that we were doing. And so that protection piece was important. And then I reached out, and it became clear that a startup company was the right vehicle. There were a couple key aspects that made this appropriate for a startup vehicle rather than my academic lab. The first was what I was talking about before, that this crosses so many different disciplines. That it required surface chemists and optical engineers and biochemists and computer scientists and material scientists, you name it. And that, recognizing the magnitude and diversity of the team that was required, that can be done to some extent in academia, but it's a little challenging gathering 30 different types of extreme experts together to work on exactly one project over a longer period of time. So that was one recognition. Another recognition was just about the scale, just the amount of resources that would be required in order to put this system together. Was beyond what is typical of an academic group. And so, and then the other was just the inventiveness and creativity and pace that might be required for bringing this all together. All of those really pointed to wanting to start with a startup company. And so the next step then was building a team and getting some resources. And so I was very fortunate. I had been involved with a number of different startup companies throughout the years, different venture funds I've consulted and advised with, as well as just had some friends. And so I reached out to my friends for advice initially and said, hey, I think this is important. I think this could have some impact. How do we, can I just get some advice so I don't mess it up? And one of the people that I reached out to was Sujal Patel, who I'd known for 15 years through his previous company, which was a company called Isilon, and our lab used a lot of their services, and reached out to him, and he got very excited. And he had been working with a venture firm called Madrona, and he himself was an angel investor. And he had been looking for something really important and impactful to be part of. And so I think one of the lessons there is sometimes unexpected things come from your network. So I sat down with Sujal, we walked through things at a whiteboard, spent a couple of days going through everything, and left that meeting with a co-founder and a CEO and a first check. So to answer Ed's question, that was where our first funding came from, was really that conversation, as well as Zad Nazem, who's now on our board. He was someone who I'd worked with and was a friend, and he was very excited about contributing in this space. And so learning that it's okay to ask for help. I think that was a big lesson, that there are other funding vehicles. There are people who really want to help do important things, and that different vehicles are right for different kinds of needs. So I think those were three really big lessons.
So the take-home message that I got there was, don't be surprised if your networks can be a great help in some of the difficult things. Yeah.
Yeah, and that people want to help. That people really are looking, if you're genuinely looking to do something impactful, people want to support that. The funding vehicles want to support that, because ultimately, if something is truly impactful, it likely can have a commercial upside, and so that is totally aligned with their goals.
So raising the funds was not all that difficult, or was it particularly just an amount of time and effort that you had to put in to secure it?
I think our most our most recent fundraise last year was at the heart of the pandemic. It was actually about the first week of shelter in place. So I wouldn't call that an easy fundraise.
No.
But I do think that the elements of being successful in that space were starting with something that was important, and then being able to articulate why it was important really clearly, being able to articulate why it was different, and then being able to articulate why this was the team that made sense to commercialize it.
There is a question here that also goes back a little bit earlier to the earlier question, Parag. It comes from Neil, and it's about if you could expand on the multiple probes on a single platform with the aim of trying to detect single proteoforms, or whether there are groups of proteoforms that you would be carrying out. How much detail can, say, 5 or 6 different scans with these family atypic or group typic affinity probes get to?
Yeah, so I mean, it's a binary expansion process. So if you use 5 probes and you have that target different aspects of the same proteoform, then potentially you can elaborate 32 distinct proteoforms in 5 cycles. And so, to Neil's point, having an atlas of all of these different proteoforms would be tremendous. It would help us know what things were unlikely to exist and what things we should go looking for. And then this platform might help us to elaborate the heterogeneity, the molecular heterogeneity of proteoforms, which I'm really excited about. I think there's a lot of uncertainty about just what is the quantitative molecular diversity at that scale.
And how many times can you use the chip to be reprobed? eventually with the washing step, if it's not covalently linked, it's going to dissipate. But if you've got covalent linking, what are you finding? Is it like 20, 30, 40 scans that you're still getting robust data from?
Yeah, so our goal ultimately, so I didn't answer this previously about how many cycles does it take to deconvolve the whole proteome. The answer there is about 200 to 300 cycles are required, which is far smaller than the 20,000 that you would have to do one at a time. And so the broader goal is to have, and we've done experiments where we've, press go and let it go for hundreds of cycles. So that's certainly something we're always optimizing. But yeah, ultimately the goal is to be able to do hundreds of cycles, and it's a non-destructive process. So potentially you could take a chip and study it and then come back to it later and reinterrogate it again.
Yeah, if you have a hypothesis that you wanted to ask, you could reprobe. That's one of the good things about the old biochip strategies that were taken in the early 2000s is you could go back and reprobe the chip. I've just had a bit of a brain fart. I was thinking about what would be the next thing that you would ask. I think going from that, the next thing I would ask would be, okay, if you're going to commercialize such a platform that is disruptive and brings lots of new data to the table. I know what I was going to remember. It must generate millions of gigabytes of information. The XY platform has a single molecule base to it then, and then you're doing that. I gather it's with things like fluorescence or chemiluminescence or something like that, right?
That's right, fluorescence.
And then each scan gives you a completely different set of data.
That's right. So it generates a gargantuan amount of data. So ultimately, the full-size chips have about 10 to the 10 landing pads for measuring about 10 billion molecules. The goal of that really being able to achieve the dynamic range that's needed in our field. And so that generates terabytes of data. So that is absolutely something that people have become more comfortable with. If you'd said, oh yeah, I'm generating 2 terabytes of data, 10 terabytes of data 15 years ago, people would have laughed you out of the room. But now genome sequencers routinely generate those scale of data. This is probably, a factor of 10 on top of what a typical genome sequencer does, but it's not unimaginable. It sounds like a large number, but it is tractable today in a way that wouldn't have been tractable 15 years ago.
Yeah, so it won't scare off collaborators and partners, which is such a big part of commercialization success.
Yeah, absolutely. And I think that's a huge point, is as we look at proteomics as a field, we need to recognize, and one thing I think we've really learned over the last many years with all these different technologies emerging, is that proteomics is huge and incredibly diverse. And so there are so many different platforms that have room to ask and answer particular questions. And accordingly, even though it's already huge, our opportunity as a field is to further expand that tent so that it is accessible to the broad biomedical community. Right now, it takes a fair amount of expertise to do a proteomic study properly. Ultimately, our success as a field will be if, like I said, anybody who wants access to the proteome gets it.
Well, that's interesting. Are you able to share with us any of the potential service agreements or early access or collaborations or technology champions that you have in place already?
So I can talk about one of them. The others we haven't announced formally yet, and so I can't out them today. But we do have an ongoing relationship with Genentech. And that's been tremendously fun working with them. And we are actively soliciting partners right now. We don't have a ton of bandwidth to do everybody, but we're excited about partnerships where we have great science, great questions, and our goal with those is really to publish so that we are able to get more information about our platform and how it works and what it can do out in the world.
So it's a proof of concept at present.
Yeah.
You proved it in-house, but now you're doing it with a collaborative partner, Genentech.
That's right.
Okay.
So I want to clarify, we have not in-house measured the entire human proteome as yet, but that is certainly our goal.
Are there any health or disease challenges that you've prioritized?
So I think we're taking a pretty wide and agnostic approach. However, diseases that we know there is not a clear genetic linkage are really interesting, because that really helps speak to the power of the proteome. And so, we see this in things like heart disease and autoimmunity and neurodegenerative diseases. And if, even though people think of cancer as a genetic disorder, there's just a huge component to the proteomics aspect. So, so I think those we're not being exclusionary, but we are absolutely in favor of areas where we can show the proteome having an outsized impact.
Do you envisage at some stage announcing that you've comprehensively mapped the enormity of the human proteoform diversity? I'd like to be at the celebration when you do.
So I think that is certainly the goal. Yes. I don't know that we will be able to characterize the entirety of the proteoform diversity, but we look forward to working with Neil on that. But certainly the goal is to enable it to be possible, so that within a single run, you are able to measure the vast majority of the proteome. And so the first of those runs will be a very, very, very exciting day. And we absolutely will be sharing that with the world when we achieve that milestone.
One thought comes to mind is that given you're using affinity reagents, could you do the reverse and use mass spec as the orthogonal technology to identify what you think you've got by saying, okay, there's 5 or 6 different affinity reagents that give us positives for this group of proteins or this group of proteoforms. Can we use that as an affinity reagent and then place whatever is captured finally onto a mass spec platform to say, yes, categorically, this is what is giving us these multi-positive reactions?
So you could certainly get in that direction, right? So you could generate a column using a multiplicity of reagents and do an enrichment in that way, and use a mass spec to demonstrate that you got enrichment of some particular protein using those reagents. So that might be a way to validate a
bit like
rare and unusual proteins. Yeah.
Well, this really brings us to a question that's one that has been posed, and that's what does Nautilus see the future of next-gen proteomics looking like? With this disruptive technology, what do you actually envisage it's going to provide to the experimentalist?
Yeah, I think, just as an aspirational goal for the field, Rudy's been talking about wanting to measure the complete proteome in a reproducible manner without missing data ever since I've known him. So I think that is still the goal, is to be able to enable the research community to have access to the piece of the proteome that they want. And I think with that, what does that do? Well, if we look at pragmatically some of the challenges that we have in some of these larger cohort studies, right now they're bottlenecked by a couple things. One is just the throughput that we're able to achieve with many of the existing technologies. Just literally being able to crunch through tens of thousands of samples is challenging. So I think that's one thing that is going to be opened up by these platforms today. I think the other, again, this missing data problem, when you're trying to do machine learning, when you're trying to do multi-omic integration, having sparse data, missing data is really difficult. And DIA, of course, addresses some of these challenges, but I think just the completeness of the data cube on large cohorts will enable us to both do a better job with discovery. We know there are lots of cell surface targets that are potentially great new drug targets that we just can't see very well today. Those cell surface preps are hard and they're very low amounts of material. So I think that's the other aspect. In the biomarker space, being able to do those really large cohort studies that are required to get around the noise of real people and the dynamism of real people's systems. And then I think the last piece is really those integrative studies that we recognize that, as much as we love the proteome, there's great value in the genome, the transcriptome, and the metabolome, the lipidome, the glycome, the imagome. And so I'm really excited about being able to click together all of these different data modalities. I think that's the other thing we're really going to see as this emerging wave of technologies come about.
Are there things that researchers should be particularly thinking about as these new approaches do roll out? Like, for example, adequate controls and changes in experimental design that people should be thinking about up front?
Yeah, absolutely. I mean, I think the way you think about a problem and asking a question when you think about a cohort that is 12 patients versus a cohort that is 15,000 patients, they're very different. And so I think that does open up wider sets of experimental designs, but it also necessitates really careful thought into controls. So really, where is the source of variation? There's so many places that the variation can come. As a community, we focused a lot on pre-analytical variables, but we simply just don't know what the variation within a person across time is to understand whether what we're seeing is statistically significant or not. So I do think researchers need to think about the entire chain and history of a sample from the second it's collected to number of freeze thaw cycles to, I'm sampling 5 microliters out of a 5 ml tube to how much do I need to sample a given person over the span of a day or from a different vein or across time to understand what fluctuations are random versus what are actual signal. So I think we really need to think very carefully about how does one construct a great experiment in light of having more tools that are more sensitive and are also going to be more susceptible to confusing signal and noise.
So taking those questions or that answer and the questions around massive data being generated, do you think it's important that Nautilus support FAIR principles, the findable, accessible, interoperable and reusable data principles that have been adopted in genomics and mass spec-based proteomics so far? Is that like something you've considered already?
Absolutely. I mean, as you know, I am a huge proponent of open data and data sharing, and that's really where ProteoWizard came from, was wanting to make it possible for people to have good access to their data. And I think that we need to address a couple different communities so I think FAIR standards are a nice start, but accessible, the A in FAIR, means different things to different people. So if you're a bench biologist, having Python access to something is, maybe not quite as helpful, whereas having a web portal where you can look at the data and integrate it with other types of data becomes really valuable. So this is something we think a lot about because we do want to support the different communities, both programmatically and non-programmatically. So absolutely going to be supporting the FAIR principles, but also ideally going further to make sure that we honor the spirit of FAIR and not just the minimum requirements.
Yeah, we got actually a really interesting question from one of your colleagues, Chris. He asked whether or not the data produced from the Nautilus platform could be combined with other data sets for interrogation by other people.
Yeah, and I think absolutely. And I think that's one of the great opportunities that we have is, I think the challenges to data integration currently, as a lab that has done a lot of work in multi-omics and proteogenomics, there are some simple sounding challenges like gene names that it sounds really simple, but when you start combining proteomics data sets with genomics data sets, the nomenclature and naming and splice variants, those are all complexities that that can really trip up a large integrative analysis. And as we talked about before, this sparse and missing data problem can also trip up analysis. So it is absolutely the hope that this data will become part of a much larger data cube to really make it possible for people to do those integrative studies, not just of omics data, but of larger clinical metadata and pathomic data and others. So I think we really want it to be as enmeshed in the ecosystem of biology and biomedicine as possible.
Thank you, Parag. There's another interesting question here from John Wilson. And John asks, what's your approach to learning the pattern of the multiple reagents that you're using to ID or quantify proteins?
Yes, so I may not be able to fully answer that, but what I can say is that we do a lot of work in probe qualification to understand not just what are the on-target effects of any given probe, but also the off-target effects. And recall that our probes are typically raised against shorter epitopes. So that's the starting point of the conversation is, presumably, hopefully it binds to the thing you raised it against to bind. But then we do a lot of epitope mapping work afterwards to understand its off-target effects. So that's really the starting point. And then programmatically, there's also a set of algorithms that we call ADAPT, which starts with a prior on what we think something binds to, and then bootstraps and learns from within the data itself.
Right. And Dan Liebler, a great question. What range of sample types do you envision? Plasma, cells, tissue, FFPEs?
Hey, Dan. So I think
Thanks, Dan.
Certainly plasma, cells, and tissue absolutely are early on our list of samples that we anticipate using. FFP is going to be a little bit further down the road because, of course, if you cross-link all the proteins, getting them sufficiently uncross-linked so that you can separate them out to single molecules may be a challenge. Absolutely something we're looking into working on, but fresh frozen and even just paraffin embedded without the cross-linking are clearly easier targets. Just to put a carrot as a nugget in your head, though, one of the things we're really excited about on a longer term horizon is single cell. There's the advantage of having a single molecule platform, of course, is it is extremely sensitive. So we're really excited about extending in the future to be able to actually do single cell proteomics. It's not where we're going to start, but it is something that the platform should eventually enable.
Great answer. You just stole my next question.
Oh, sorry.
No, it's okay. Here's one from the Chromosome Human Proteome Project team. Do you think that Nautilus's platform may unearth credible proteomics data for many of the current PE2, what they call missing proteins?
Yeah, so I think there are two classes of missing proteins. There are some that are non-translational proteins, right? Things that come about through alternative creation processes. Those ones are a little challenging because essentially what the platform will do is it'll say, okay, I have a protein that binds to this set of things, but it doesn't look like anything I've seen before. And so that's where you could do something fun, like flip it around and use the platform as a really intriguing enrichment method to dig up those proteins and profile them on an orthogonal platform. So I think that's those things that have no reference whatsoever. But within the PE class of proteins, many of those, you see them in the genome, you just haven't been able to measure them as yet. And I think that's where this platform will have, I think, a lot of benefit because of its sensitivity, it being protein-centric instead of peptide-centric. So the advantages of dynamic range will be helpful there, and not having the ion suppression challenges that mass spectrometry typically faces. So I'm really hopeful that for those classes of proteins that are known to exist from the genome or from splicing, but maybe are just challenging to access by mass spectrometry platforms.
Yes, I think you know that I've got an interest in one of the big classes that's missing, the olfactory receptors. We don't have any very strong proteomic data for any of the 404 olfactory receptors, and I wonder whether there's any bias in the anchoring chemistry that you're using for membrane soluble proteins or not.
Yeah, there shouldn't be, and that actually relates to Giannis's question. Hi, Giannis. As well as I think Ed had a question about sample prep. So currently the sample prep aims to denature the proteins. It doesn't aim to keep them intact. The probes target linear epitopes, and so having denatured proteins is actually helpful. And once the proteins are immobilized, they're glued down, so you can be quite flexible with the solvents that you use in the system. So with regards to membrane proteins, you don't have some of the challenges that you have with other platforms because you're able to use pretty harsh solvents all throughout the process.
So I can tell you that there are olfactory receptor family typic sequences that you could make antibodies against that would pick up those 404 proteins, hopefully.
Well, that'll be a fun, maybe that's a fun collaboration we should talk about.
Already done a run on it. I was just looking for any other questions. Gustavo has made a comment and said, amazing achievement, congratulations. Thanks, Gustavo. Ed also asked, will you be looking at the microbiome?
So, I think there are a couple of places where it's important to recognize the limitations of the platform. And that also comes back to what we were talking about before about making sure that we are not overhyping things. And I think there are going to be a couple classes of proteomes, the microbiome being one of them, probably the immunopeptidome, and a few of these other places where there's either just no good sense of what the reference is, that will be an early challenge for the platform. Ultimately, it would be great to address those, but those won't be amongst our early targets.
And the last one we've got here, and we're still taking questions, we've got a minute or so left, is where are you at with publications, with regards to publications? Yeah,
so our target that we've shared is that our goal this year is to submit some of our seminal publications this year, and so that's really our goal, is to get those out in the world for everyone to be able to learn more about what we've been working on.
Well, thanks, Parag. It's been a revelation this morning or this afternoon for all my colleagues in the States. And Andy, I don't know whether you want to take back over now so that we can finish on time, but thank you very much, Parag, on behalf of the US HUPO community. It's been a real pleasure, and you've certainly shown us something that we think is going to, or we hope is going to, provide a lot of answers to problems that we've been facing for many years. So well done, mate.
Thanks. It's always great chatting with you, and you as well. And Andy, thanks so much for inviting me.
Thank you.
Look forward to catching up with you soon.
I feel that this interview is really unique. It's out of my expression, actually, a lot of technical details. I didn't realize that. And Parag is certainly very inspiring, talking about your vision about next generation proteomics. So I I try to have maybe personal interact with it, but I think some of the audience here probably interested. So I think a lot of us are potentially faculty members. We are interested in commercial and technologies. So can you, Parag, so can you tell us a little bit about that? So, I mean, we have another job, which is in the university, especially at the beginning. So how you at that time, maybe have time, also the visions to really put a lot of effort to commercial technology. Did you recruit your student doing that, or what's the step you do? And then later on, and I think also we have some problems with these technologies, how much you need to prove, because proteomics technique sometimes is challenging, consider sometimes not ready for commercialization. So how many steps you need to move? I mean, apparently you're very successful, moving very fast, right? So we would like to hear from you.
Yeah, well, so I think there are a couple of pieces there. About one is just the early phases, and it's sort of a deep question to understand when is the right time to commercialize. That something that you've been working on in your lab, it's maturing, you're de-risking it, and there. My suggestion there is to be in contact early with potential investors. Many people wait till the point where they want money and they're like, okay, I need money next week. And instead, it's much better to bring them into the journey and share with them and say, here's where we are. Do you feel like this is something that is ready, or are these particular risks still barriers? And so having that conversation early and often, not in a I'm trying to raise money, but just in a I'm trying to keep you informed and part of the journey, is really, really important. I think that's one huge lesson that I learned, is that you don't wait till you need money to start chatting with the people who have it. And then with regards to balancing academic and industry, there are a couple places where that can get really, you have to be careful. One is particularly if you have active development in your lab alongside active development in the company, the conflict of interest there has to be navigated very carefully. And Stanford is very rigorous about this, which is fantastic, to make sure that you're not abusing your graduate students and forcing them to do work for the company. And I'm really glad that they're as rigorous as they are. So I think what I found is that both of them are really important experiences. And in terms of finding balance, thinking about what does the company need you for? What is the value that you're providing to the company, and making sure that you're doing that. And also having the humility to recognize that, there are things that there are people who are out there who do way better than you. Sujal is amazing. And so I am so grateful to him every day, because he has experience and knowledge of all of these commercial trajectories and commercialization and product development. And he knows how to think about building a large publicly traded entity, that I just don't, I don't have those experiences. So having that humility to get the right people around you and enable them to do their job, rather than feeling like, oh, I'm the professor, this needs to be mine. I think that is a huge piece of success, as well as it helps you balance your time most efficiently. Thank you.
Yeah, that's certainly absolutely important. Yeah. I wish I could ask more questions, but I think we have to stop here, and thank Parag and Mark again. I think the whole proteomics community is very, very much inspired by the Nautilus for sure, and the vision for how we move forward with the technologies, right? Yeah, and thank you again.
Thank you, Andy, for the chance.
Yeah, thanks so much.
Thank you. We thank everyone for today, and I hope you guys will join us for next US HUPO Global Campus event. Bye-bye.